Second one, hybridization of selected parents. Third, selection and evaluation of the hybrids. Fourth, testing and release of new varieties. Single cell protein is one of the alternative sources of proteins for nutrition of humans. So the variety is Pusa spermi, Pusa A4, which is resistant to what? Hello everyone, welcome back to session 5 of this very important chapter called Strategies for Enhancement in Food Production. What did we learn in the last session? We studied about plant breeding, that is mutation breeding, and also we studied about Dr. M.S. Swaminathan, who is the man behind the Green Revolution, right? In today's session, what are we going to study? So, plant breeding for disease resistance is very important. So, with the technology of plant breeding techniques, we are able to produce a crop which gives more yield and disease resistant. So, as we know, many of crops are destroyed by the insects. So, insects pest is something like a very challenging for the cultivars. So, producing the crops which is disease resistant is a challenge, right? So, in today's session, we are going to study about what is the plant breeding for disease resistance. As you know, many of our crops are affected by fungi, bacterial and viral pathogens which is going to destroy the whole crop. As you know, crops are very important for the economy of the country and they need not only the quantity of the crop is to be maintained, even the quality of the crop also need to be maintained, and as well as we know, for the farmers the yield also is very important. So imagine if a crop is infected with a fungi or a virus or a bacterial pathogen, definitely it is going to decrease the yield and it is going to affect the farmer. So let us see as to what is this. As I said, here you can see the beautiful picture here, right? So here a wide range of fungal, bacterial and viral pathogens affect the yield of cultivated crop species, especially in the tropical climates, particularly in the tropical climates. So in this situation, imagine in the tropical climatic conditions, most of the crops are affected by fungal, bacterial and viral pathogens. So in that case, so definitely breeding and development of cultivars resistant to disease enhances the food production. As I said, production food, it is going to affect the economy of that particular crop. So as we come across many diseases like the brown rust of wheat, red rot of sugarcane and late blight of potato. By bacteria we come across a black rot of crucifer, and by viruses you might have come across tobacco mosaic virus, or what we call it as TMV virus, and the turnip mosaic virus. So all these, this is a challenge to produce a crop which is disease resistant. That is the main aim of this plant breeding or mutation. So what is it? The very important aim of plant breeding is what? Plant breeding for disease resistance. As we know that a wide range of fungal, bacterial and viral pathogens affect the yield of the cultivated crop species, especially in the tropical climate. So in that situation, what they have to do? The breeding, that is in this situation, breeding and development of cultivars resistant to disease is very important. Then only it is going to enhance what? The food production. So before breeding is undertaken, it is important to know what? The causative organism that causes that particular disease. Then proper care can be taken to avoid the infection by that particular disease causing organism or a disease causing pathogen. So we come across a good number of diseases like by fungi, like what we call it as the brown rust of wheat, red rot of sugarcane and late blight of potato, and by bacteria we come across black rot of crucifer, and by viruses you might have come across the tobacco mosaic virus, and in turnip it is called turnip mosaic virus. So what are the methods of breeding for disease resistance? First one is called screening germplasm for resistance sources. Second one, hybridization of selected parents. Third, selection and evaluation of the hybrids. Fourth, testing and release of new varieties. So all these are the methods of breeding for disease resistant. So screening of germplasm for resistant sources. So as you know, genetic variability is the root of any breeding program. As we know, genetic variability and the entire collection of the plant species having all the diverse alleles in a gene in a given crop is called as what? Germplasm collection. Next, hybridization of selected parents. So cross hybridization of two selected parents is mainly achieved by emasculation and bagging to produce the characters of both the parents. To produce the characters of both the parents, that is called hybrid. Then selection and evaluation of the hybrid. So here selection is done from the progeny of hybrids produced by cross hybridization. Fourth very important method is testing and release of new varieties. So selected pure lines are evaluated from their yield. So all these four are the very important methods for disease resistance. One is called screening germplasm for resistant sources. Second one is hybridization of selected parents. Third one, selection and evaluation of the hybrids. Fourth, testing and release of new varieties. Plant breeding for developing resistance to insect pests. Very important. As I said, what is the main aim of plant hybridization technique? To produce a crop of which gives more yield and which is disease resistant. Particularly, as I said, the very important trait for plant breeding is what? Tolerance to insect pests is very important. Developing a crop which has the tolerance for insect pests is very important. So another major cause of large-scale destruction of plant and crop produces insect and pest. All of us know that most of the crops are destroyed by the insects and the pests. So what do we call it as the pest, particularly pest infestation. So insect resistant in host crop plants may be due to, and as we know, depending upon the crop variety, the crop may be resistant or may be prone to these insects or pests, and may be due to morphological, biochemical or physiological characteristics. For example, like hairy leaves in several plants are associated with resistance to insect pests. For example, resistance to jessids in cotton, they are resistant because they have what? Hairy leaves. So this is one advantage for the plants to resist the insect pests. For instance, like in jessids, in cotton and cereal leaf beetle in wheat. So they have what? They have hairy leaves. So that is one method of adaptation to these insects. And in wheat, solid stems lead to non-preference by the stem, soft light and smooth-leaved and nectarless cotton varieties do not attract bollworms. You might have come across the stem borers. The stem borers. So they are going to go deep into the stem and suck all the nutrients of the stem and cause a disease. So particularly there are some like smooth-leaved and nectarless cotton varieties which do not attract the bollworms. High aspartic acid, low nitrogen and sugar content in maize leads to what? Resistance to maize stem borers. So all these are some of the techniques that is developed by plant breeding. Plant breeding for developing what? Resistant to insect pests. Moving across the breeding methods that involve the same steps for any other agronomic traits. So here will come across the type of a crop and the variety and the resistance to which insect pests. So first one is called Brassica, that is the variety is called Pusa Gaurav and it is resistant to what? It is resistant to aphids. The second one called the flat bean, which is the varieties of two types. One is called Pusa Sem 2 and Pusa Sem 3, which is resistant to jessids, aphids and fruit borers. We come across the insects in the form of stem borers or even the fruit borers. The other one called Okra, Vendi. So the variety is Pusa Swarni, Pusa A4, which is resistant to what? It is developed in such a way that it is resistant to shoot and fruit borer. So all these are the crops which are developed through plant breeding techniques to overcome the insects. Moving across the next very important concept called biofortification. coming across biofortification. Very, very important concept. As we know nowadays, the diet what we have is lacking many of the micronutrients, vitamins, and even iron, zinc, etc. etc. So the problem is that diets are lacking essential micronutrients, particularly like what? Like iron, vitamin A, iodine, zinc. So what will this deficiency of this micronutrients lead to? Definitely it is going to lead to the occurrence of many diseases. Particularly, as I said, it will increase the risk of diseases and reduce lifespan and reduce mental ability. So the only answer for this is what? Biofortification. So what is biofortification? Breeding crops with higher levels of vitamins. So when you develop a crop which is having higher levels of vitamins and minerals or higher protein and healthier fats, then definitely it is going to compensate the loss that has been caused due to the lack of those important micronutrients in the diet. So what is biofortification? This is a very important term which is asked in the exam. Right, what is biofortification? What are the objectives of biofortification? So breeding, what is biofortification? Here you can see the different types of cereal crops that are produced here, right? So all these are what? Very, very important proteins. You know, the pulses are very rich in proteins. So when you develop a crop which is very rich in protein and all other important micronutrients, then definitely the person will not be having a risk of being prone to diseases. So what is biofortification? Breeding crops with higher levels of vitamins and minerals or higher protein and healthier fat, not the fat which is going to increase the cholesterol level and block the blood vessels. It is a healthier fat is the most practical means to improve public health, and this is called as what? Biofortification, right? Now let us see what are the objectives of biofortification. What is the main aim? As I said, when the diet is lacking most of the micronutrients, it needs to be compensated. So it is through biofortification. So biofortification is undertaken with the objectives of improving what? As I said, not just the quantity of the yield is very important. Even the quality of the yield also is very, very important. Quantity of the food that is developed through plant breeding also is equally important, not just yield. Yes, of course, yield along with quality also is very important. So protein content and quality. So what is the main objective of biofortification? To improve the crops having more protein content and also maintain the good quality of the protein. Then oil content and quality, very, very important. Then vitamin content and micronutrient and mineral content. So the main objective is what? To retain all the micronutrients in the crop, in the food crop, and even the quality and the quantity is also maintained. So here the protein content and quality. Second one, oil content and quality. Vitamin content and micronutrient and mineral content is very, very important. Now let us study. So what are the main objectives? This is very important from examination point of view. So usually this is asked in the examination. That is, what is biofortification and what are the objectives of biofortification? Now let us study some of the examples. So maize hybrids rich in amino acids like lysine, tryptophan compared to other varieties. So you develop through a plant breeding techniques or through biofortification, you develop a maize hybrid which is rich in amino acids like lysine and tryptophan compared to the other crops. Next, wheat variety, that is Atlas 66, having high protein content. All these are the staple food, protein, even the carbohydrate, all these are the staple food which we have in our daily life. But here the quality of that carbohydrate and protein content should be better quality. So the wheat variety, that is Atlas 66, having high protein content. So one more, iron fortified rice which has more iron as in commonly consumed variety. So developing the crops in such a way that it maintains all the micronutrients and all the protein content, vitamins, all that has to be maintained in the food that is produced. So iron fortified rice which has more iron as in commonly consumed varieties. Then vitamin A enriched carrot, spinach and pumpkin. So develop such type of crops which is rich in vitamin A. like it might be the carrot. All of you know that carrot is very rich in vitamin A, then even the spinach and pumpkin. Then vitamins C enriched bitter gourd, then mustard and tomato. Then one more, iron and calcium enriched spinach. The other one is protein enriched beans, that is broad bean, lab, French and garden peas. All these are the examples for this biofortification. So try to remember all these very important examples. Moving on to hidden hunger. Moving on to the hidden hunger. All of us know that around 3 billion people are suffering from the lack of micronutrients. So people suffering from micronutrient, that is protein, micronutrients, proteins and vitamin deficiency, is called hidden hunger. So what is hidden hunger? That is the people are suffering from the deficiencies. It may be a deficiency of micronutrient or protein, or it might be vitamin deficiency, and that is called as hidden hunger. And moving on to the single cell protein. So particularly, as we know that more than 25% of human population are suffering from hidden hunger, and definitely the single cell proteins are coming to the rescue of such people. So what is single cell protein? SCP, short form is what? SCP. Single cell protein is one of the alternative sources of proteins for nutrition of humans and other animals. So as I said, many people are suffering from a deficiency of vitamins, may it be of micronutrients, protein. So the alternate means is what? It is through a source is what? That is single cell protein. So single cell protein is one of the alternative sources of protein for nutrition of human and other animals. So here the proteins are obtained from what? Unicellular cyanobacteria like spirulina. So what is another way you can define SCP? That is production of protein biomass in large scale using microorganisms and growing them in low cost raw material is called single cell protein. So very important concept from examination point of view. So what is hidden hunger? As I said, around 3 billion people in our country, are the human population are suffering from hidden hunger. So what is hidden hunger? People suffering from lack of micronutrients, proteins and vitamin deficiency is called as hidden hunger. So in order to, as an alternate means of source is what? Single cell protein. So what is single cell protein? Single cell protein is one of the alternative sources of proteins for nutrition of human and other animals. Proteins are obtained from what? From unicellular cyanobacteria like spirulina. Here you can see, right? You can see this beautiful picture. Single cell protein. Now, what are the advantages of single cell protein? So, in order to compensate the deficiency, or in order to compensate the lack of micronutrients and proteins, as I said, SCP is the main source. So, what are the advantages of the single cell protein? So, single cell proteins are rich in proteins, as I said, minerals, vitamins and carbohydrates, and low in fat. So, they can be taken as what? Alternate means of sources of all these content. It may be protein, minerals, vitamins and carbohydrates. Next, they can be easily grown using cheaper materials like waste from potato processing plants and animal manure and molasses. As I already said, growing them on low cost raw materials may it be like waste from potato processing plants or from animal manure, molasses, etc. Then the use of waste materials also what? Reduces what? Pollution as culture medium. So they are using what? The waste material as the culture medium, and obviously it is going to reduce what? Pollution. This is very important. Then they reduce the pressure on agriculture. What for? For supply of pesticides, fertilizers, etc. So that is also very important. Example, 250 gram of methylophilus methylotrophus bacterium has been used to produce 25 tons of proteins. Imagine the amount of proteins, single cell proteins that are developed through this bacterium called Methylophilus methylotrophus. Around 250 gram of this bacteria produces how much? 25 tons of proteins. Imagine the amount of protein that is developed through the single cell protein. So all these are the very important advantages of single cell protein. Hope you have understood all the important concepts what I have explained in today's session. So I'll be back in the coming session with one more very important and interesting concept called plant tissue culture. So I'll be back with, as I said, with some more concepts of plant tissue culture. Till then, goodbye and thank you.